The last part of Module 4, Cellular Localization, predicting where your protein is found in the cell, is a tool called Phobius. So you're going to go there. Phobius is going to pull together as a confirmatory second tool. It's like a double check built into this whole project here to find where your protein is in the cell. So it's kind of like HMM and signal P together. So what you do is going to be fairly similar. So we need our FASTA sequence again, no problem. And you want to do long with graphics and then submit your query. Pretty easy. And it's very quick spit out. Okay, so dissecting this a little bit, the red line is the probability of this protein, KSED00010, being a signal, having a signal peptide. And you can see that that red line is at a 0. That means that this protein is very likely not secreted from the cell. It stays inside it somehow, or it's in the membrane. So the next thing, is it in the membrane? The blue and green lines indicate portions of the protein that are non-cytoplasmic, meaning they're extracellular, or cytoplasmic, which means they're intracellular, respectively. So blue, extracellular. Well, this is actually an artifact. This is your likelihood that it's extracellular. And cytoplasmic, you can see it's above 0. If you do have helices in TMHMM, you might see that they don't match the numbers or the counts here in Phobius. So if that happens for you, go to page 95 in the training manual for cellular localization data, and you can definitely find out what's going on there, and it might help. Some of these tools cut the length of your amino acid sequence down to the most likely area where you can find these things. In the case of mine here, there's just this little blip of what they're saying is a helix, but it's not very significant. It would be a hydrophobic section of the protein. But I'm going to want to grab a picture of this, so I'm going to just type down here snip, whoops. And I want to get everything. When you save it to the place where you're keeping all these pictures. You're going to want some pictures for your poster, especially if you have something cool that comes up. A great picture that shows transmembrane helices or a signal peptide is really a great idea for a picture to pick. Phobius probability graph. I am going to browse for it, and I saved mine to my desktop. Remember, you can stretch it, double check it's there, and it is. Save it. And as far as a hypothesis, this is where you're going to look back at your tools that you used in this module. So here at the beginning of cellular localization data, we did the gram stain. We did a little basic research, which is great to use on your poster. TMHMM, we predicted no transmembrane helices. That means it's not likely to be in the cell membrane, mounted in the membrane, which is hydrophobic on the interior, and hydrophobic helices would embed in the membrane very well. So we saw that, not likely to be transmembrane helices, and we concluded that here. With signal P, we were looking for a signal peptide, which would signal that protein to be secreted outside of the cell, and we found that no signal peptide was predicted. And we had a graph for that. It did not reach the threshold that we needed. LipoP didn't apply because for KSED00010, it's gram-positive. LipoP would apply if you had a gram-negative bacteria. P-sort B gave us a really high cytoplasmic score. P-sort B said cytoplasmic, yep, very high. And then Phobius was telling us that, well, it doesn't have a signal peptide, and it's not likely to be transmembrane at all. So they really all kind of confirm each other. So this is either going to be cytoplasmic. So when you go back for your hypothesis, no transmembrane helices are predicted for KSED 00010. Nor is the signal peptide predicted, therefore the protein is most likely cytoplasmic. So at this point in this module, I have now established where it likely resides, what its name probably is, and function has come from other modules, and I am ready for the poster. And I would just say I got done through module 4. So if you got all that stuff done, that's a great job. Do a nice job with your poster. I'm here to help. Thanks.